Hello and welcome to the Immune Epitope database. My name is Nina Blazeska and I'm the Project Manager of the IEDB. In this demonstration video, I'll be showing you a step-by-step process of how to access the ImmunomeBrowser for B-cell data for SARS-CoV-2 epitopes, focusing on the spike protein. To start, we'll locate the epitope source panel on the IEDB homepage and select our organism of choice. So here I'll type in SARS, and as you can see, a series of related organisms are populated in the dropdown based on community ontologies. Now I'll select SARS-CoV-2. You're also able to select an antigen, but for the purposes of this tutorial, we will keep this blank. Next, we can select our assays of interest, and for this tutorial, we'll be looking at B-cell assays. You're also able to select your assay outcome of choice. So positive data means when in the assay, the B-cell recognizes that epitope and generates a response against it, whilst negative means the B-cell does not generate a response against that epitope. So to understand the ImmunomeBrowser better, we'll select both positive and negative. And lastly, as we are focusing on SARS-CoV-2, we'll only be looking at human data, so I'll select that in the host panel. I'll keep all my other parameters the same and hit the green search. And as that's loading, you can see that the B-cell view has been selected in our filter options based on our original query, and all my other query parameters are populated at the top. And now you can see all of the carefully curated B-cell data available for SARS-CoV-2. As you can see, we have many different tabs with various results. So the epitope, antigen, assay, receptor, and references tab. And you can also export your results here. In the next step, we'll be focusing on the ImmunomeBrowser, which can be found by clicking on the Antigens tab. And this is just a pop-up reminding us of where the ImmunomeBrowser can be found. So as a result, I'll click on this histogram-like button. And now this is taking me to the ImmunomeBrowser. So at the top here, we have the name of the protein we are looking into. So SARS-CoV-2 spike glycoprotein. Next to it, there is also a link to the UniProt database, which has the protein sequence data, which we used as a reference to align with our protein. As you can see, there are also two graphs here of interest. So let's talk about this first graph, response frequency, or RF. So this is a graph informing us of how immunogenic a specific residue is in the analyzed protein. So RF equals R on N, where R is the number of subjects responded and N is the number of subjects tested. The x-axis here shows the values that are the residue numbers of the protein, while the y-axis indicates the response frequencies. What we see here is actually a confidence interval of the response frequency. This is very important because it allows us to correct for the sample size. Otherwise, a peptide found to be positive in only one donor, so R equals 1, N equals 1, would be considered more immunogenic than a peptide showing response in 99 out of 100 donors, as the former has an RF score of 1 and the latter of 0.99. So this graph is showing a range of possible values that will contain the real response frequency value 95% of the time if the same experiment is repeated an infinite number of times under the exact same conditions. Of course, we'll have different values for B-cell and T-cell data, so it is expected that the response frequency will differ even if we compare this with the same protein. So you can also export the results that are used to plot this graph here, and you can download or save the data in various file formats. Moving down to the epitope assay counts graph. So this shows how many times a specific amino acid is present in a positive or negative assay. So this graph gives us an idea of the immunogenicity from that region. And similarly, you can also download or save this data. Important to note that blue line indicates the positive data, whilst the red line indicates the negative. And then moving down to our table, this table shows every epitope that has an assay related to it. So that first column, epitope ID, contains the IEDB accession number of the epitopes, while the second column contains the exact epitope sequence. And as you can see, we have both linear sequences and discontinuous sequences. In the third column, we have the mapped position, which indicates where that epitope is located in the reference protein. And this is followed by the percent sequence identity between the epitope and the corresponding region in the reference protein. The next columns relate to subjects tested, responded, and the assay outcome. And lastly, the last column is the response frequency when we have a confidence interval of 95%. Now I'd like to dig a little deeper. We'd like to see what regions in the SARS-CoV-2 spike glycoprotein are the most immunogenic. So I'll just hover over the graphs here, but residues 13, just around here, to residue 303, which is around here. This comprises the N-terminal domain. Next, I'd like to zoom in on a particular area. So residues 319, which is just around here, to 541, which is just below this area, zooming in here. This makes up the receptor binding domain, or the RBD. Within the RBD, there's a particular area called the receptor binding motif, and this is from residue 437, which is just about here, to residue 508, which is just around here. And now I'd like to zoom back out. From these two plots, we see that the RBD domain is just one such area of interest. And apart from this, there are a few other areas that are fairly immunogenic. So again, I'll hover, but some examples that we have, residues of 530. So we're looking at around about here to residue 600. That area is quite immunogenic. We see from 612, which is just around here, to 659 here. That area is quite immunogenic, and in fact the residue D614, which is frequently mutated in the variants, is located in this immunogenic region. And then lastly, region 760, which is about here, to 840, about there. That is another immunogenic region. So next, we'd like to see where mutations are observed in some of the circulating SARS-CoV-2 variants within these immunogenic regions. So to make this a bit easier, I'll be zooming in on a region about here to here. So for example, we have at position 484, we have a mutation E484K, which is observed in the following four variants: P.1, B.1.351, B.1.1.7, and B.1.617. And this is obviously situated in a highly immunogenic region and can thus alter antibody responses. Similarly, we have another frequently occurring mutation at position 501, which is just around here. And this mutation is N501Y, which is present in variants P.1, B.1.351, and B.1.1.7. Again, another highly immunogenic region. I have a few other examples to show you. So at position 452, we have L452R mutation, which is present in B.1.617, which was first detected in India, as well as two variants here in California, B.1.427 and B.1.429. And this is all, of course, at the time of recording this video. And then lastly, at position 417, we have mutation K417N, which is present in the B.1.351 variant, first detected in South Africa. So hopefully these examples demonstrate how one can use this information to assess whether the mutations in the variants are likely to affect the antibody responses to SARS-CoV-2 by determining whether they are located in highly immunogenic regions or not. As this demonstration, we hope, has shown, the IEDB can provide some really useful information about currently available SARS-CoV-2 data, which can be analysed to obtain some important insights. As more data continues to be deposited into the database, the results in the ImmunomeBrowser will, of course, grow richer. We also aim to add some new features to the ImmunomeBrowser in the future to continue enhancing that user experience and of course, the usefulness of these graphs for your research. But for now, we hope you enjoyed this demonstration, and thank you very much.